everyone, welcome back. And I'm looking a little different right now. So I'm working on a new technique, and I want to introduce it. So bear with me. I'm working here with this thing down here. But the background I put up because I'm really a little bit homesick, and so it's my cousin sent me this picture from the mountains back home, and so I just put it up because I like it. The way it looks into the mountains is just sort of pretty. But anyway, what we're gonna do today is I'm gonna bring this up big like that, as big as I can make it. Let's see if there's a little bigger. Oop, yeah, there you go. And today we're gonna talk about molecular biology. And when I look at molecular biology, right here, molecular biology. And let's see, did it put it? Look at that, it's doing it right. And I can even do this. Oops, there we go. Put this down, there we go. So what is molecular biology? Molecular biology is how to make protein. So how to make protein. There we go. Isn't that nice? That way I don't have to worry about my handwriting so much. So how to make protein. When we look at how to make protein, it's an assembly line situation, and we need to have some kind of an instruction place of where we understand how to assemble the proteins. Because when we look at protein, it's the amino acid after each other, and how we're going to figure out which amino acid to put after one another. And that's sort of the process of what we're talking about today, a little bit elaborate, because the first thing we need to do is, where is the cookbook? Where is the instruction manual? The DNA is sort of the foundation of it all. And DNA stands for deoxyribonucleic acid. Deoxyribonucleic acid. That's a complicated way. Deoxyribonucleic acid. Look at that. It spelled it right. That's good. That means my handwriting is not that shabby, I hope. So what we are going to do now, we're going to look at that whole thing a little bit more. The DNA was discovered in the around 1950s, or thereof, when it was really sort of established of how this is how it looks. So the discovery, we call it around 1950s. Okay, and so what then, when we look deeper into the DNA structure, then we go very fast and very quickly, we get us into nucleotides. And we've already touched on those a little bit, but let's now go deeper into talking about the structure of those, and then how they are assembled, and then from there we can go and talk about how we make the proteins. All right, let's talk about some of these nucleotides. So we're going to have us the nucleotides. And the first thing we're going to do is we're going to talk about the structure of the nucleotides. So let's see here, structure. I'm going to talk about the structure of a nucleotide. What is it made up of? And when we look at that, we have three things that it is made up of. The first thing we got is a deoxyribose sugar. And you can see when you look at that sugar here, that is a 5-carbon sugar. So we have these carbons. See, actually what we can do is these carbons, we can zoom in a little bit here. See, we can number them. They always number. There's an oxygen, and then we have these 5-carbon sugars. 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon sugars. And so that's a deoxyribose sugar one. And then the other thing what we have there, oh wait, I should write this down. We should write this down here. 5-carbon sugar. And so as you can see, they don't draw the sugar, they don't write the carbons. That's just where the connections are. But they write the other ones, so you know the other ones, what they are. So the 5-carbon sugar is there. And then we have as a phosphate group attached to it. The phosphate group is, the phosphate group is over here. And then the other thing we also have is a nitrogenous base, and that's on that side, the nitrogenous base. And so this just says base. So what we want to make sure is we understand. See all these nitrogens that is attached there? Look at that. I could do this. See all these nitrogens that go here like that? So that's the place where nitrogenous come from. So we want to make sure we understand that. Nitrogenous. Those bases are. So there we go. All right, well, let's look at those a little bit more, because we actually have 4 different types that make up and the different nucleotides. So we actually have 5 different nucleotides, but in DNA we use 4, and then in RNA we exchange 1 and use the other 1. And so that makes it 5. And so we have these adenine, guanine, thymosin, and cytosine. We have those, and then in RNA we also have one that's called uracil. We're going to talk about that when we get to that. So the 4 nitrogenous bases can either be 1-ringed structures with nitrogen in it, or 2-ring structures. So what we have here, we have some of them, as you can see, have this 1 ring here, and then the other one have 2 rings here. We call them different things. We call the 1-ring structure the pyrimidines, and the 2-ring structures are the purines. And what's going to happen is, this is what's going to make the variety of 4 the nucleotides. The phosphate part is going to create a backbone. We're going to talk about that in a moment. And then on the other side, so when we look at, so the phosphate part here, that part is the backbone. And then this part is the variants, that we have 4 different parts. And these are like tags sticking out. And when you look at the DNA molecule, the double rung structure, you have these tags sticking out, these rungs. That's what these things are, and they're going to attach with one another. And those are going to attach with one another via a hydrogen bond. And that's like a zipper. You can zip it, unzip it, and close it again and open it again. But the backbone here, those are covalent bonds, and those are very, very strong bonds. And so when we look at then the nitrogenous bases, we have these 1-rings and these 2-ring structures, and one of each is going to connect with the other one together. So what we're going to end up having, and we learned that before, we're going to have a purine and a pyrimidine come together. We're actually going to have the the cytosine connect together with the guanine, and then we're going to have the thymine connect together with the adenine. And those are then the ones that can connect and create those rungs. So that's very, very specific. Now, they can either be on either side of the rung, so we get a fair amount of variability coming up. But it's always that those 2 go together and those 2 go together and connect with one another. All right, so that's good for now. We're in that, and let's move on now and see what happens when we connect those nucleotides to one another.